Walaikum Assalam. Yes, Razan, how are you? Alhamdulillah. Okay, let's just start the class. Uh, the last class we did learn about the natural vegetative propagation methods. Today we are going to continue our discussion from the artificial vegetative propagation methods. Okay, now what is artificial vegetative propagation and what are the different methods of this artificial vegetative propagation? Okay. Before that, Razan, do let me know if you have any questions. Okay. Also, your voice is coming very low. You're hardly audible to me. Okay. Meanwhile, let me. Okay. Okay.
Can you hear me?
Yeah, you're audible, but not that properly. It is kind of low. Voice is very low.
Let me change it to headphones.
That will be better.
Sir, can you hear me now?
Yeah, you're audible. Okay, let's start the class then. Others still have not joined. Okay. Razan, by the way, turn your camera on in the class. So far in the class, we have learned about different natural methods of vegetative propagation in which there is no intervention of humans. There is no role of human to play in that. Today, we are going to learn about artificial vegetative propagation, which will be carried out by we humans. Either it will be done in the field, in the agricultural fields, or either it will be, some of them will be done in the laboratories. Okay. So some of the most common type of vegetative propagation that is carried naturally, that is carried artificially, I meant to say, are grafting, cutting, layering, and tissue culture. These are some of the most common artificial vegetative propagation methods that we carry. Getting it? So that is the case of artificial vegetative propagations, and some of the common examples we had as follows. Common types. The first one here we have grafting, then you have cutting, third one is layering, and tissue culture. These are some of the common types of artificial vegetative propagation. I am putting the screenshot in the chat box. You can note it down from there. Yes, Fatima, Assalamu alaikum. How are you? Yeah, 1 minute. In the chat box, I can't see the names of you guys. 1 minute. You guys just give me a minute. Actually, I'm trying to put this. I have to put the screenshot of the notes in the chat box, but there I can't see your names. So I will be quickly connecting to the team, and then we're going to continue. Yes, Razan.
For the last, is it tissue culture?
Yeah, it is tissue culture. It is tissue culture. TISSUE. Okay, na. Let me quickly make a call. I will be continuing in a minute. Okay. Something needs to be resolved here. I will be back. also. इस आईडी से मैंने ज्वाइन किया ना, एक अदर आईडी से सुहेल नाम से, कि स्क्रीनशॉट्स शेयर करना होता है चैट में, लेकिन मैं वहाँ पे बच्चे का नाम भी शेयर हो नहीं रहा है वहाँ पे। हाँ, अभी शो हो रहा है, अभी हो रहा है। ओके, थैंक यू। Yeah, now I can see you guys. Okay. To everyone, yeah. Okay, continuing further. Yes, Aman, how are you trying to join the class on the given time? Everyone? Yeah. So these are some of the 4 common types of artificial vegetative propagation which we are going to learn about in the class today. Okay. Also, the screenshots I will be from now on, I will be dropping it in the chat box. Okay, now in the previous class, I also think I dropped it, and you guys will be noting it down from there after the class. So the first type of artificial vegetative propagation we are going to learn about is cutting. So in the method of cutting, we will take a part of the plant. Specifically, we take a part of stem, or in some cases, we take part of leaf also. But majority of the plants, we will take the stem, and this stem will be planted in the soil. This stem will be planted in the soil. But the important thing is that, say for example, if you were to take this plant here, suppose this plant you have, and say in order to cut this stem and plant it in the soil, a new plant will grow out of it if it contains nodes in it. If it contains nodes, then only a new plant will be growing on it, growing out of it. But if it does not contain nodes, then new plant will not be growing. Getting what I'm trying to say? Take for example, if I were to cut out this much part of this plant, let's say I have cut out this much part of the plant, and if I am going to sow it in the soil, if I am going to put it in the soil, a new plant is not going to grow out of it. Why? Because node is not present in this. Getting Fatima, Razan, and Aman, everyone?
Yes, sir.
Okay. Razan, rename your correctly, I suppose. Yeah, yeah, rename yourself correctly. So nodes needs to be present on the stem. That's why a new plant will be growing out of it. Okay. So further, like you can see, when you look at the stem of the bamboo here.
Sir, how do I rename myself?
Okay, you're not getting that option. Okay, let me quickly rename you. Okay, done. So what I was saying that, like this plant here, as we have taken the stem, so we have to make sure that the nodes should be present here. So cutting plant with node point in the soil. Okay, na. Also, what you will be doing, suppose this part of the plant has been cut, upper part of the plant has been cut. So this one will be put into the soil first, not the upper part. It's an obvious thing. And say here how the bamboo will be growing. So these are the different nodes present on the bamboo tree, on the bamboo plant here. Okay, and as you will be putting it in the soil, so roots will start to emerge from it. Okay, these are called as adventitious roots. Okay, from these cuttings, adventitious roots will start to develop. From this one also, special roots called as adventitious roots will start to emerge from the side of the stem, and hence a new plant will grow then. By the way, you guys know that a grass is not a tree. Huh? Why grass is not a tree? If it is not, I mean, just sorry, I was asking about this bamboo. Why bamboo is not a tree? What is it? Do we consider this as a tree, or do we consider this as a grass? Any
Sir,
idea to
we
anyone?
consider it as a tree, right? Because grass is very thin.
No.
We consider it,
Grass?
we
Yeah,
consider this
grass.
as a leaf. I mean, grass.
Grass, yeah. It is
Yeah.
considered as a grass, okay, na? Because it has several characteristics that makes it different from the trees. Right now, there are several characteristics of the bamboo plant that makes it different from a tree. That's why it is considered as a type of grass and not a tree. Okay, there are several reasons for it. That is one thing. Okay. Also, the bamboo's stem is called as culm. You might see CULM is written here, Actually, the bamboo's stem is especially called as culm, C-U-L-M, a term used for it. So you guys understood what is cutting and how it is done? Yeah. So we basically take a stem of the plant and put it into the soil, but we need to make sure that the nodes are present on that stem. Only the new plant is going to emerge out of it, right? So sometimes, I mean to say, this is the way how it will be done. Okay. And the examples of the plants that will be grown in this manner include, we have rose plant, we have bamboo plant. These are some of the examples. Can you guys think of some other examples of plants that can be grown by the cutting method? Yeah. Think of examples of some plants that can be grown by the cutting method, other than the ones which we have mentioned here. The very common plant, we have sugarcane.
Yeah, sugarcane.
Exactly. Sugarcane is also grown by the cutting method. Okay. Other examples.
There's sunflower.
Sunflower.
Yeah.
Sunflower, I don't think sunflower is grown in this manner. I don't think. Okay. Sunflower, I'm not 100% sure about it. But generally, I don't think it is grown in this manner. Okay. No?
Okay.
Other examples, we have examples of like a rubber tree can be grown in this manner.
So rubber tree.
Exactly.
So the rubber tree method is actually very cool.
Yeah. In fact, it is very useful plant. The latex that we obtain from it is used to make rubber, different kind of objects.
It kind of looks like a coconut, but it's different.
Yeah, exactly. It looks like the pulp of the coconut, the white edible part of the coconut, but it is very different. Okay. Like when it is stored in that, when it is stored now, it becomes very fluffy and very soft.
Yeah.
So we have rubber plant. So that's all about the cutting method. As you guys can see here, as this cut part of the stem has been put under the soil, so you will see that roots will start to emerge from it. Okay, no? That is the case here. Another method is the grafting method. So sometimes what happens? Sometimes two plants will be joined together so that both of them can provide the desired characteristic to a new plant. Both of them can provide desired characteristic to a new plant. Okay. Most commonly, two different types of mango plant or two different types of apple plant. The one for two different parts of the apple or the mango plant will be joined together to obtain a new plant that will have desired characteristics that will be very favorable. Okay, no? So there will be several advantages of grafting. We have several advantages of grafting. Like when we will be obtaining a new plant by the method of grafting, it is observed that they will be resistant to some diseases. Okay, now they will be able to bear the environmental stress. If the environmental conditions are not very good, even then those plants will be able to sustain, will be able to survive in that unfavorable environmental conditions. We'll be learning about advantages. Let's get back to the main thing here. So in this, we take the stem from some plant, and it is like we have taken the stem of one of the plant, and it has been attached to the stem of another plant that is rooted into the ground. That is attached into the ground. Getting it, guys? So this part, this stem, it is rooted in the ground, and you have taken the another part, and you have taken the stem from another plant. Both of them have been joined here. There are different ways of joining it here. We have made a cut like this. Okay, now one of them has been cut into an arrow shape, and one of them it has been cut into the V shape like this. Okay, now and it has been simply inserted into this, and with the help of some cloth or some tape, it will be binded tightly. Okay, sometimes we can use some growth promoting chemicals also. Sometimes growth promoting chemicals can also be sprayed on this to keep it safe from infection, or to trigger the growth, or to allow it to grow properly. So after some time, what will happen? The tissues of these two plants will be grafted together. The tissues of these two plants will integrate It will integrate, it will join, and it will act as one plant. Both of them will become as one plant after some time. So that is the case here. So one plant remains rooted in the ground, and this is called as the stalk. And this stalk will be providing the essential nutrient and water. Its function will be to provide the water, essential minerals, or basically the nutrients, right? And the other one is called as scion, right now. So other one will be called as scion, okay, the one that you have attached to it, okay. So in this way, we are developing a new plant, and the new plant will be providing you desired characteristics. Meaning, for example, the new plant that will be growing now. Let's suppose that we have taken two plants here. Let's say we are taking example of mango, let's say. To say that the stalk is good in the sense that its water absorption capacity is very good. Okay, now it's a very really good absorber of water, right? But the fruits that it bears, it's not of very high quality. Say, for example, we have initially two plants. Try to understand this. Okay. Okay, we have taken the mango example. Okay. Let's say we have no example of apple plant, for example. Let's say. Now, when you look at the stalk of this plant, no, when you look at this plant, it is good in the sense now stalk is good absorber of water. But when we look at the fruit that is being beared by this plant, it is not red, and neither it is sweet. Okay, now, so let's say that apple that is growing on this plant, it is sour and it is green. So is it a favorable, is it a good characteristic of the apple? Is it a good thing? It's not very sweet, and it is plus green, let's say. It is not, it will not be very profitable for the farmer to sell in the market. Now take the other plant here. So this one is the scion, no? The upper part. Now in this case, the apple that is growing here, it is sweet, plus it is red in color also. Getting it, everyone? But the problem with the stalk is now, the stalk is not good absorber of water.
Sir, can I go drink water?
Okay, okay. All if you pay attention here, what I'm trying to explain here, you have two different plants. Both of them are of apple, but of different variety. So the plant A here, let's call this as plant A, let's call this as plant B. So one of the like the stalk of this plant has good absorption of water. The roots are good absorber of water. Right, Razan, Aman, you guys getting it, everyone? But the apple that is growing on it, it's sour and it is green. Take the other plant here. In this case, the upper part of the plant, the scion, the apple growing on it is sweet and it is red. But the root system is not good. The root system is not good. It's not a good absorber of water. So both of them are having one plus point, one negative point. There is some pros, there is some cons about these plants. Getting everyone what I'm trying to say? Razan, getting enough? Fatima, you back? Aman? Is Aman you there in the class or no? So what we are going to do now, we are going to take the root system of this plant, basically the stalk, which is a good absorber. We're going to take this part and we're going to take this part and we are going to combine both of it. So we have taken the stalk and we have taken the scion, and both of them have been joined together here by the method of grafting. So the new plant that will grow, its root system will also be good. 1 minute. Its root system will also be good now. Plus the fruit that will be growing on this will be of higher quality. So that is the purpose. That is why grafting is done. This is the reason behind doing this method. Is that clear to everyone?
Yes, sir.
Okay. Fatima, you're not back in the class?
Yes, I'm. Yes, sir, I'm here.
So you understood this. So we
Sir,
have
could you
to,
explain it again?
we are saying that we had two plants, plant A and plant B respectively. The root system of plant A is very good. Okay, now they are very good absorber of water. The root is wide and it grows to very much. It grows to more depth in the grows deeper into the soil. That is a good plus point. But the fruit that is growing on this plant, let's say, it is not of very high quality. It is sour and it is green, let's say. So that makes it less favorable. When you look at the other plant here, the root system is not very good. The root system is not good, but we are seeing that the apple that is growing on it is sweet and it is red. Okay, now it is sweet and red. So
Okay.
what we are doing here, we have taken this lower part of the plant and we have taken the upper part of the plant of the same variety here, of the same type but of the different variety. So we have taken the scion and we have taken the stalk. Both of them have been grafted here. So the new fruit that will be growing will be of very high quality. Will be of very high quality. Getting it, everyone, now? Because the new plant that will grow, its root system will also be very good. Its fruit will also be very good. Right now, that is one thing. And in fact, in a single plant, several varieties of fruits or vegetables can be grown. Right now. Also the new plant that is going to grow, it is resistant to different kind of diseases. It is resistant to different kind of microorganisms, right? And that is one thing. And the growth of the plant will also be very fast now. Like you don't have to wait for almost an year to get fruit from the new plant. Getting my point now? Like if you were to grow the plant from the seed, if you want to grow the plant from the seed, it will take almost about 1 or more than 1 year to get the fruit, right? Depending upon the plant also. But in this case, you don't have to wait for about a year or more than that to get the fruit. That is only. Also, we are able to grow fruits or vegetables of different variety on one single plant, right? That is one thing. Also, like grafting can be used to cut off damaged parts of the plant also and replace them with new tissues. If suppose some part of the plant has been damaged, let's say this part of the plant has been damaged, so I can simply cut it and I can graft a new stem to this part right now. Any questions from the method of grafting? Please let me know. Yes.
Like, do we use grafting to fix a plant? Like that?
Rizan, speak a little bit louder.
Do we use grafting like to fix a plant?
Yeah, we can use it. Okay, now it's not the very common method. I mean to say, it's not in every situation you can use, but yeah, it is one of the option. Say for example, if one of the part of the plant has been damaged, not the whole plant, I'm saying. Says one of the branch is being damaged. Okay, now it has been infected. So we can simply cut it off and we can graft a new stem to this part of the plant. We can do the grafting in that manner. Okay, that is one thing. So let us understand what were some of the advantages of this. So in a small bracket here, I'm going to write down this advantages in short. The first advantage is that it offers resistance towards different kind of diseases. Okay, so the new plant will be called as grafted plants, and grafted plants are resistant to many diseases. Another advantage is that it grows faster. You don't have to wait for a year or two in order to get the fruit. So it faster growth is seen in such kind of plants. Right, that is one thing. Other inputs, I mean to say, other benefits about grafting would be like, for example, as we discussed moments ago, it can be used to repair the damaged parts of the plant. Okay, we can repair damaged parts of plants. So that is the case. And there are several examples of plants that are grafted. The common examples are mango. You have apple. You have guava. Uh, there are several examples. In fact, plants like roses, they are also grafted. Plants like roses, hibiscus, they are also grafted. So after this, we have some examples. We have mango, apple. In the flowering plants, you have the example of a rose, hibiscus, rose, hibiscus. You have example of guava also. That's it. I'm going to put the screenshot in the chat box. After the class, you guys will write it down in your copies, okay?
Okay, sir.
Also
Sir,
important,
like, should we draw the tree as well?
you can just draw one of the trees, like just this one. Let me point it. Just draw this one. Okay, no?
Okay.
This one and then this one. Don't draw this one.
Okay.
Okay, no? Also important thing to be noted down is that the tissues of these two plants now, they will integrate, they will mix and act as one. So they will become single plant after some time. Earlier they were two different plants, but after some time they will become a single plant. That is one important thing. Next, we have the method of layering. So in this method, what we're going to do, we are going to forcefully bend the stems of the plants towards the ground, and then we'll be covering with soil. As you guys can see here, with the help of the metal rings, the stems of the plant has been forcefully bent towards the ground, and later on it will be covered with soil. And from the part which has been covered with soil, a special type of roots called as adventitious roots will start to emerge from this. Adventitious roots will grow, and then finally the shoot system, as you guys can see here, the shoot system also starts to grow. So that is the method of layering. Okay, now that is called as layering. And this stem, let me highlight this here. This stem that is attached to the main parent plant, now, that is called as layer. So that is being called as layer. Okay, so hope it should be clear. Any questions from this method?
Can you repeat like the layering again?
Repeat again.
Can you repeat what layering is?
Sure. In this method, what we have done here, we have taken one of the stems of the plant, okay, and now we are bending it towards the ground with the help of these metal rings. As we can see here, these are metal rings. We are going to kind of hammer it into the ground so that it will hold the stem close to the ground. Also, we'll be covering it with soil, and we will see that roots will start to grow from it. What kind of roots will be developing? These are special roots, and we call these as adventitious roots. Adventitious roots from which a new plant will emerge. So adventitious roots have grown here, plus the shoot system has also grown. The shoot system too has grown from this. So that is the concept of the layering. And this attached stem in which the roots are developing, new plants have grown, this whole attached stem is called as layer, Layer. Is that clear or not?
Yes, sir.
Okay, no? So that was the method of layering, which is a artificial vegetative propagation method. Aman, you there in class? You getting the concept or not? Now later on, what we can do? Now we can cut off the Now we can cut off the stem from the main plant, and all these plants will start to grow out separately, right? Now, like for example, each plant that have grown here, we can separate it. We can just cut them, and all of them start to grow individually, grow separately, right? Now, so important point thing to be noted down is that later all the new grown plants can be cut from the parent plant, and they grows independently. Earlier, while they were still attached to the parent plant, earlier while they were still attached to the parent plant, when we have not cut them, so they will be getting nutrition from the parent plant also. Getting it now? But once you have cut and separated them, so the new plant will be obtaining nutrition from the soil. Okay, or you can take it from here and you can plant it in somewhere else also. So some of the common examples of plant that will be grown by the layering method are as follows. So we have examples like raspberry. Raspberry is one such example of plants like jasmine. Yeah, these are some of the examples. We have other examples like strawberry is also one such example. Strawberry is one example. Then you have grapes also. So these are some of the common examples that are artificially vegetatively propagated using the method of layering. If there's any question in this, please let me know. Good. Okay, I forgot to put the screenshot. You guys are saving it in your devices or not? Make sure to save all the screenshots.
Yes, sir, I'm saving it.
Good. Next, we have the method of tissue culture. So in this, we can collect cells or a small part from any part of the plant. We are going to take a small fraction from any part of the plant, and we will take it to the laboratory. In the laboratory, what we are going to do, we are going to put it under a glass, and in that glass, we are going to use some special chemical substances that promotes the growth. So for example, you have collected a tiny tissue sample from this plant here. It could be from any part of the plant. Okay. Now we have put it under a special medium that promotes the growth. And how does this special medium will be promoting growth? It contains nutrients and growth promoting hormones in it. Growth promoting hormones in it. So the first step is to collect the tissue sample from the parent plant. Next, you will be putting it under a glass structure like this. In this glass structure, we have put some special substances. So some special chemicals are put in this, and those special chemical substances helps in promoting the growth. It will help in promoting the growth. Okay, now. So also they contain nutrients in it. So they contain nutrients and promotes growth. So the first step is this, and this is the second step which we are discussing in detail. So we have put the sample of the tissue in this. After some time, not on the same day, but after some time, we will see that after a few weeks, we will see that those from that tissue sample now, tiny plantlets have started to grow. Plantlets are very tiny plant. Very tiny plantlet, very tiny plants are called as plantlets. So that has started to develop. Now later on, as these plantlets will be growing, their requirement for the nutrient is going to increase now. So we are no longer going to grow it in this glass structure. Okay, now we are going to put it into the compost. Compost is basically a type of manure. It's a kind of a fertilizer. Okay, now we have to add manures and fertilizer to the soil, you guys know, to improve its fertility so that plants grows in a healthier manner, more nutrients are supplied to the plant. So compost is basically a type of fertilizer which is rich in nutrients. Okay, so there you will be now transferring it to there. So the plantlets will, those tiny plantlets will now be put into the compost and there it will grow. That is one thing. Is the steps of tissue culture clear to everyone or not?
Yes, sir.
Everyone got it? Aman, you, and Fatima, you also. So here we have been able to grow an entire plant from just a small piece of tissue from the plant in a laboratory. Okay, now that is one thing. Sometimes this tissue culture, it is also called as micropropagation. Micropropagation is also the term used for this. Micro here refers to something that is really very small in size. Something that is very small, it is called as micro, right now. So thereby it is also called as micropropagation. A complete new plant is being produced, is being made from just a tiny fraction of the plant part here. That is something. Also, in this, are we in this, are they growing under the natural conditions, or are we trying to mimic the natural condition for the growth of this plant here?
Natural conditions.
I mean to say here, are we growing it in the natural conditions? No, it's being grown in a laboratory now, in a laboratory right now. So for what kind of plant tissue culture will be done? So those plants which are not able to grow under the natural habitats, under the natural conditions properly, or they are endangered species of plants, right now. So there are several species of plants that are endangered. You guys know what is endangered species of plants, right? So if the population of some organism is very less, if it is decreasing, and if it is and if the decrease of the population of that organism is not controlled, in the nearby future it can become extinct from earth. So they are called as endangered organisms. Getting it, guys? Oh. So we have examples of endangered plants and endangered animals both. Okay, talking about the endangered plants, particularly we have the example of like Malabar lily is one such example of endangered plant. Or you have example of ebony tree. It is found in parts of India also. Umbrella tree, that is also. So these are the trees that are endangered, and if we were to grow them in the natural conditions, it will take lots of time now, because the growth will take lots of time, plus they might not be able to grow in the natural conditions properly. Okay, so thereby we'll be preferring the method of tissue culture. Also, you can see that how many plants are we are able to grow from just tiny fraction. So at a given time, we can grow about hundreds to thousands of plants from just one tiny fraction of the plant. Is that thing clear to everyone?
Yes, sir.
Fatima, you getting it?
Yes.
Okay. If anyone has any questions in this, please let me know.
No, sir.
Okay. Another important thing is that we have to see the examples of the plants that are grown via tissue culture. Although majority of the plants can be grown via the tissue culture, but if a plant is not an endangered plant, so it does not make any sense to grow that plant via the tissue culture method. So tissue culture method will be usually used to grow those kind of plants that are endangered. Getting the point now, all of you?
Yes, sir.
So we have examples of plants like you have umbrella plant. Have you guys seen this umbrella tree or umbrella tree? Umbrella plant, have you guys seen this? After the class, now Google about this. You will see that this plant, its leaves will be growing in the shape of an umbrella. Okay, no? So that is one thing. Other examples include the ebony tree. etc. Now further the question is like, what are the advantages of this? So one advantage of the tissue culture is that we can grow the plants in a very short span of time. Other advantages include like we will be able to protect the endangered species of plants now. So some of the major advantages of tissue culture are as follows. First thing is it saves the time, plus we can save endangered plants. Endangered plants can be saved in this manner. Okay. Apart from these, there are several other importance of this. We can grow the plants that will be disease free. That will be free from the disease and the different kind of viruses. Okay, na? So disease free plants can be grown by this method. I hope this much should be clear, and this should be clear to everyone, I suppose. If any question, please let me know.
No, sir.
Okay, I have put the screenshot in the chat box. So we have learned about all different methods of artificial vegetative propagation so far. Okay, now, in fact, prior to that, before that, we have learned about natural methods of vegetative propagation also. But nonetheless, the thing is that if you were to talk about all of them together, whether it be natural or whether it be the artificial, all of them comes with some advantages. So together, whether we talk about the artificial or natural vegetative propagation, the plants that will be produced by the vegetative propagation, they will be growing faster than those that are produced with the sexual reproduction. Because in the case of sexual reproduction, Razan focus here in the class. In the case of the sexual reproduction, the new plants are growing from the seeds now. So that will take more time, isn't it? Further, the plants that will be growing by the vegetative propagation method, they will be bearing fruits and flowers ahead of time. Okay, now, so before time they will be bearing the fruits and flowers in it. And the plants that are being produced by the vegetative propagation methods, they have the same characteristics as that of their parent. So if the parent plant produces a very rich quality fruit or vegetable, so as the new plant will also do it. Okay, that is one thing. So these are some of the importance of the vegetative propagation. We can maintain the quality of the plant. Okay, now we can maintain the quality of the plant here. That is one thing. If anyone has any questions, please let me know.
No, sir.
Right. Now we guys know that there are some plants that are seedless, that does not have any seed in them. You guys know there are some plants, like some fruits that we consume, they don't have seeds in them. Like for example, pineapple. Have you guys ever seen a seed in the pineapple?
No.
Yeah, Fatima,
Some
Aman.
oranges.
Yes, sir.
Some oranges they don't have seeds, right?
No, oranges do have seeds in them.
No, like some oranges don't have.
They have, but they have a very small size seeds. But they you're saying that some oranges don't have seeds.
Yeah, but I have ate like my father used to bring
Okay,
seedless oranges.
I suppose you're talking about some of the oranges like navel oranges. There's a species of oranges. Okay, na, that does not have seed in it. I suppose the name of the orange thing you're talking about, it's navel oranges. Okay, let me write it down. NAVL, navel orange. This is the orange. As I know, this is the orange. One of the orange that does not have any seed in it. In fact, it is a very large size orange. Okay. Getting back to the question, we were talking about the seedless plants. Yes,
But,
Razani,
the seedless orange I ate is small size,
small size. I was saying that it can. I think it can be big. It can be small also. Okay. Right. Now, talking about one of the advantage of vegetative propagation is that there are several plants now. For example, pineapple. There are some seedless grapes also, or plants like banana. They can be grown only via the vegetative propagation method only. Getting my point now? They can be grown only by this method only. There's no other option or alternative for that plant, right? So seedless plants like pineapple, pineapple, bananas. We have pineapples, and then we have bananas, and we have examples like dahlia, and we have some other examples. Like for example, you can take there's roses also. Rose can also be taken as such example. It does not have any seed in it. It does not produce the seeds. So roses, pineapple, banana, dahlia, right?
Sir, what
So
about
they can
corn?
yeah,
Corn.
corn.
Seed.
No, corn is not vegetatively propagated. You're talking about corn, CORN, right? Right, Razan?
Yes, sir.
No, corn is not a seedless plant. Okay, no. They themselves are the seeds. Like corn, basically, they are the seeds now. Okay, no. So corn is not a seedless plant. These are some of the seedless plants that can be grown only by the vegetative propagation method. Can only be grown via vegetative propagation. Okay, so these are some of the advantages. Next, we have some special type of asexual reproduction. We have budding and we have fragmentation. First of all, let's learn about the budding here. So there are some organisms that are just made up of one cell. Take for example, yeast. So yeast is an organism that just contains one cell and it is a fungi but not a plant. Many times, yes.
Plant organisms that have one cell are called unicellular.
Exactly. We call them as unicellular organisms right now. And the organisms that have more than one cells, they are called as multicellular animals, multicellular organisms. So the reason why we are studying about budding here, usually as we are talking about budding, so budding is observed in the case of fungi. And fungi is not a plant. Many times we confuse fungi with plant, thereby we are discussing about this. Okay, now many times students and people confuse fungi as a plant, but it is not a plant. That's why we are learning about how the fungi does reproduction, merely because of this confusion. So what happens here? As you guys can see here, this is a yeast here. And a small bud has grown out of this. Right now, a small tissue grows out of it that is being called as bud. Right? And after some time, it can detach from the parent plant, from the parent organism, I mean to say, or it can remain attached. So this is the parent yeast from which a small tissue has grown that is called as bud. Right now, so later on it will grow, it will multiply further, and a new yeast cell has grown out of it. Now it can separate from the parent plant. I mean to say
Sir,
parent organism. Yeah.
what does a parent yeast means?
What does the term
Yeast.
parent yeast? Basically, like the parent. This is the yeast from which a new organism is going to grow. Now, so it is being called as the parent of the new yeast. Right now, it's the parent, just like in the case of humans, we talk about parents in that sense. So in the similar manner, this term is being used for the yeast. Okay, now, so it's the parent yeast from which the baby yeast is going to grow in that sense. So a small outgrowth, that is bud, has grown out of it. The bud will start to grow further and it will grow into a new yeast cell. Later on, it can separate from the parent yeast. but in some cases it can remain attached to the parent yeast, and there will be a chain of yeast cells that will be attached to each other. That is one thing. So this method of growth is called as budding, which is another asexual method of reproduction. Also, this can reproduce very quickly if it gets the favorable conditions, if it gets the right amount of nutrients, if it is available to it. So within few hours, it can propagate, it can spread very quickly, right? That is one thing. Okay, no? So this is all about the method of the budding, as you guys have learned here. We have some other special organisms also which does reproduction via budding. Again, this is not related to the plants because the animation that you are seeing, this is of a microscopic animal. Okay, now this is a microscopic multicellular animal. It is being classified as an animal which also does reproduction via the method of budding. In this, what happens? There will be a small outgrowth from the body. From any part of the body, there will be a small outgrowth, and this two here is being called as the bud. And later on, it will grow and it will finally detach from the parent organism. It detaches itself from the parent organism and will start to grow separately. So that is an important thing. If there's any question in this, please let me know.
Sir. We're teachers.
I'm going to put the screenshot of this one also. Okay. By the way, this is the Hydra. I didn't told the name. This is Hydra. Okay, now. Next, we are going to learn about fragmentation. And fragmentation, in this generally some of the alga, for example, Spirogyra is an alga. What happens, how it will be doing reproduction? Basically, it will split into multiple parts. It will split into different fragments, and each fragment will grow into a new Spirogyra, right now. So what happens that many times you guys would have seen that in the water bodies now, in the water bodies, the alga will grow uncontrollably. It will grow in a rapid manner, and its growth will be very difficult to control. So when this alga like Spirogyra, when it gets favorable amount of, when it gets enough amount of water and nutrients, then its fragment will start to grow into new individuals, into new organisms. As we can see here, this Spirogyra has split into two parts, and these two parts, both of them are growing individually. Both of them have grown into a new Spirogyra. So that was the parent Spirogyra here, which is splitted into two new Spirogyra, Spirogyra 1 and Spirogyra 2, we can call them like this, and they are growing independently. And this can grow in a very short period of time. It can multiply very quickly in a short period of time. So that's all about fragmentation, right now. So alga like Spirogyra, they will be generally reproducing by the method of fragmentation, in which they will be dividing themselves into several parts, and as they will be getting enough amount of water and nutrients, all the fragments will start to grow into new organisms, and they will further multiply very quickly. In a very short period of time, they will multiply quickly. Any questions from this? Please let me know.
No, sir.
Fatima and others also tell me.
No, sir.
Okay, no? That is one thing. Okay, I forgot to put the screenshot. Next, we have the spore formation. Okay. Now, as you guys can see here, that the example which I have taken here, it is just to make you understand in detail about the structure of the spores here. Okay. So this is the image whenever we take a look at the bread now, and after some time, if the bread expires, so you guys will see that brown and greenish patches will develop on it. Might have observed this, right?
Yes, sir.
Fatima and Rasan here. So those are actually fungus that grows on it. They are actually fungus that is growing on it. Now, when we look at the structure of the fungus in detail, when we study about them, so we find that they are structures like this. Structures like this. Both of them are same structure basically here. On the top of it, there's a bulb-like structure that is called as sporangium. What do we call this? Yes, Fatima and Rizan, that is sporangium. Okay, now you guys can see here, this sporangium has exploded. This is exploding, and from this, thousands of tiny spores have started to come out. Thousands of tiny spores are being released from the sporangium as it will be bursting, and wherever these spores will be landing, if it finds favorable condition, if it gets moisture and nutrients, from there it will start to grow into a new fungus. Is this thing clear?
Yes, sir.
But no. Yes, Razan?
Yes, sir.
But the question is why we are learning about fungi, because fungi is not a plant now. We are learning about it because it will be helping us to understand how even some plants can do reproduction with the help of the spores. So they are plants like fern and moss. Look at the leaf of the fern here. These brown structures that you see, they are, can you guys make a guess? What are these structures here? Yes, Fatima and Razan, what are they? They are the spores now.
There was this Purani. What's the name, sir?
What's the?
What's the name? Like you just told.
It's the fern, F-E-R-N. It's written here, by the way. Fern and the moss here. This is the fern plant and this is a moss plant. Okay. So that is spores, S-P-O-R-E-S. Okay. So there are some plants that contains spores on their leaves particularly, and they will be basically floating in the air. Basically, leaves will be floating in the air, kind of, right? And spores, they have a hard protective cover outside their body. When we look at these spores now, they're actually kind of hard. They have a hard protective cover on top of them. So hard protective cover is surrounding it. And this hard protective cover on the spore will be protecting it from unfavorable conditions, meaning that even if the environmental conditions were to change, if the temperature and humidity were to increase too much, this spore will be able to survive. It will not be contaminated, it will not rot, right? And since they are very lightweight now, so with the help of the wind, they can be carried from one place to another. They can travel long distances with the help of the wind. And as soon as they will be landing to the favorable conditions, to a favorable place, suppose if it lands on a very fertile soil, this spore were to land on the soil, so now it will start to grow into a new plant now. A plant will start to emerge out of this. This was the spore which has landed on the soil. So as it will be landing on the soil, just how the seed germinates, this will start to first germinate and a new plant will start to grow from it. Is that thing clear to everyone? How with the help of the spore also, plants can do reproduction? Let me know if it is clear or not.
Yes, sir.